Chapter 6, Segment 3, pages 176 through 183. Through evolution, vertebrate brains have changed in both size and organization. All vertebrate brains appear to have a spinal cord, a hindbrain, midbrain, and a dicephalon and a telencephalon that has a cerebral hemisphere, along with other regions found in typical mammalian brains. The lamprey has the most primitive features of all living vertebrae. A lamprey cerebellum is very small compared to other vertebrae. Birds and mammals have a large cerebellar hemispheres that evolved independently from a reptilian cerebellum. Increased size of cerebellum may be due to increased complexity of sensory processing and motor agility. The difference in brain structure between the many different vertebrate species is the relative size of the brain structure. All mammals have a six-layered cortex, which is known as a neocortex, which comes from the Greek word neo, which means new, and the Latin word cortex, which means the bark of a tree. In the most recently evolved mammals, more than half of the brain's volume is made up of the cortex. The cortex is mainly responsible for higher order functionings, for an example, perception of objects. Midbrain optic lobes and midbrain optic centers that were responsible for perceptual functions in lesser developed mammals are presently visual reflex centers in modern mammals. Reptiles were the first vertebrates to possess a cerebral cortex, as well as the first vertebrates to exhibit large cerebral hemispheres. Reptiles only have 3 cortical layers, which mammals have 6 cortical layers, as we've discussed, neocortex. Reptiles, which is an archaic cortex, and that means 3-layer cortex, comes from the Greek word arche, which means ancient. The cortex of reptiles may be homologous to the 3-layered hippocampus in mammals. Brain size evolved independently in multiple lineages. There is much variation in brain size within each line of evolution when we compare animals that are roughly the same body size. Hagfish are the most recent members of an ancient class of jawless fish, and they have the forebrains that are 4 times that of a lamprey, with a comparable body size. Increases in brain size is related to the behavioral capacity most completely in animals. The encephalization factor. A relationship between how body size and brain size are related was first found in modern species, but then it was successfully applied to fossils. In the 17th century, human beings discovered that we do not have the largest brains as we previously thought. An elephant's brain is 3 times as much as a human's brain. Later on, though, whale brains were discovered to be even larger than elephants' brains. As a result of these findings, scientists suggested that brain weight should be considered only a fraction of the body weight. This guaranteed that human beings outranked elephants, whales, and other large animals. But when we take this into consideration, we still aren't the smartest human beings. We aren't the smartest mammals. Mice have almost the same ratio of brain weight to body weight as humans do. Figure 6.13. Across various species, there is an overall correlation between body and brain weight. This is why all points on the plot fall within a narrow polygon. Linear regression is a statistical procedure that creates a line of best fit, which passes through, or closely through, the marked points, which represent various mammal species. The regression line shown in figure 6.13 has a slope of about 0.69. This represents the mathematical relationship between body weight and body weight across all mammals. With this figure, you can compare how much an individual species deviates from the expected range. Deviation, or residual, corresponds to the vertical distance for the species above or below the diagonal line on the graph. The distance, k, is known as the encephalization factor. The greater the k for a species, which means the higher above the diagonal line, the larger the brain size for that species, which was more than originally thought it would be. Patterns of connectivity in gene expression may have more importance in distinguishing the human brain from the brains of related species, compared to the gross anatomical features, for example, size and cell count of the brain. Brain size can be the result of evolutionary adaptive pressures. This wasn't true for dinosaurs, though. Brain size as adaptation. The size of certain brain regions is linked with specific abilities. For example, navigating by sound, foraging for food, performing a large repertoire of songs. Some capabilities may have generated a general increase in brain size. These capabilities are related to overall cortical volume, rather than the volume of one specific region. When the brain increases in size, normally most major parts of the brain will increase in proportion to the overall size increase. Even with regards to the gross anatomical level, there are differences in rates of increase. For example, some species have a relatively larger olfactory bulb. This is presumed to be the result of the pressures of natural selection favoring a highly developed sense of smell for these particular species. Across various species, we have observed that the medulla becomes proportionally smaller relative to brain weight, the cerebellum stays proportionate to the brain weight, and the cortex becomes larger than any other part. Over the course of primate evolution, the brain regions that have increased the most are the ones involved with the more complex functioning and develop later in life. The human medulla is fully developed at birth, but the cortex will continue to add neurons throughout childhood. Theorists propose that larger brains evolve by prolonging the later stages of development. This pattern can be a possible explanation to the changes in the fine structures of the cortex. During fetal development, the innermost layers of the cortex are the first to develop, and then neurons are added to create each outer layer. It can be seen across various mammals that the outer layers 1 and 2, of the cortex, that were added afterwards, have increased more in primates than in innermost layers. Many factors led to the rapid evolution of a large cortex in primates. Hominin, primates within the family Hominidae. This includes 4 genera of great apes, one of which being modern human beings. Hominin brains enlarged rapidly in our recent evolution. Australopithecines had relatively small brains that were about the size of modern chimpanzee brains. Approximately 10 million years ago, trunks and arms become their modern form. Over 4 million years ago, we become bipedal. 2.5 million years ago, fossil records indicate the manufacturing and use of stones, tools, in daily lives. Australopithecines made the unutilized sharpened stone tools as a means of hunting and butchering. The creation and utilization of tools lessened the need for large jaws and teeth, and this resulted in a steady decrease in size over time. Approximately 2 million years later, the last of the Australopithecus species developed modern species. Dexterous hands, smaller teeth, and a trend toward increasing cranial capacity. Homo erectus. As Homo erectus evolved, the brain steadily increased in size as the face steadily decreased in size. We made stone tools, could start and utilize fires, and killed large animals. Fossils and tools of Homo erectus have been found on 3 continents, whereas Australopithecine remains were only found in Africa. 150,000 years ago, Homo sapiens appeared, and they had the modern brain volume. Australopithecine's brain volume remained basically unchanged after 2 million years, but in 1.5 million years, the brain of the Homo species had almost tripled in volume. The brain has now been altered in size since Homo sapiens first appeared, possibly because of the consequences of having a large brain. Negative and positive selection pressures affected hominin brain size. Large brains result in a large gestation period and difficulties when giving birth. It's especially an issue for bipeds due to the large brain of our species having to pass through a narrow pelvis, which the Australopithecine evolved so they could balance over 2 feet rather than 4. After a baby is born, the brain still continues to grow, which results in prolonged dependence and prenatal care towards the infant. Chimpanzees' brain weight levels off shortly after the birth, whereas the human brain continues to grow at a rate that rivals that of the growth of the fetus. Most of the postnatal brain growth is due to the expansion of the cortex. Even though the human brain only makes up 2% of our body weight, it consumes a greater proportion of the metabolic intake. The human brain's construction and maintenance is very complex, that over half of our genes are devoted to these tasks. Mutations of any of these genes can lead to behavior disorders. Changes in organs over the course of evolution is usually because this change offers a fitness advantage, that is, an increased likelihood that an individual will survive and reproduce. One hypothesis suggests that the increase in brain size is due to selection pressures in the social domain. Across primates, there is a correlation between the average size of the social group and the size of the cortex relative to overall brain size. The social brain hypothesis suggests that a larger cortex is needed to aid an individual in maintaining relationships with other large-brained individuals. Correlations between social group size and brain size indicate that the possibility exists to estimate the maximum size of the species' social groups via the size of the cortex. The ratio of cortex to the brain may be an indicator in the number of meaningful relationships we can have with other individuals. Another view of the increase in brain size is due to 3 different factors: innovations in behavior, the use of tools, and social learning, which is learning by observing others. A study correlated brain size in 116 species of primates with these 3 factors and used not only the total brain weight, but also the ratio of executive brain, which is the forebrain, to the brainstem. The total brain size, as well as relative forebrain size, had a positive correlation with regards to each of the 3 factors. This indicated that each factor is related to the increase in the size of primate brain. Brain size predicts success in adapting to a novel environment. Brain size correlates with how complex feeding behavior is, as well as with the social group size with regards to primates. Hypothesis: enlarged brains evolved as a more general adaptation to allow us to cope with sudden environmental changes. If we were to test this hypothesis, one way would be to introduce a species with a large and small brain into new environments and see if they were able to adapt to the changes and thrive in the environment. This would be an unethical experiment, though, because of the unpredictable environmental problems that arise when a non-native species is released into a new environment. An alternative way of testing this hypothesis would be studying the past record of human-mediated introductions of non-native species into the environment. A comprehensive study utilized a global database to study more than 600 examples of the introduction of 195 different species of birds into the environment. This study found that species with larger brains compared to body size tend to be more successful in adapting to the new environment.